Thank you. So thank you to the committee for inviting me. So my name is Jérôme, and I work at INAIA with my supervisors, Fabien Souchanek, Bertrand Thirion, and Gaël Varocco. And I'm going to talk about our work about extracting information about the brain from the neuroimaging literature, and more specifically about finding the statistical link between the text that appears in a neuroscience publication and the brain regions that they are describing. So I'm not going to introduce neuroimaging because we've heard a lot about it today. Will be the data that we use in my work is mostly journal articles based on fMRI studies, functional MRI studies. So these tools are used to measure the activity of the brain, and it enables researchers in cognitive science to study how the brain implements certain mental functions, like retrieving a memory or recognizing an object, or to study how pathologies affect the functional organization of the brain. Unfortunately, many of these studies are conducted with few subjects and lack statistical power, and sometimes their findings can also be specific to a particular experimental setup that was used during the study. So we want to be able to pull together many studies and perform meta-analysis to find out which links between mental function or pathologies and brain structure are reported consistently across studies. Also, the aim of this statistical analysis of many published articles is to provide a high-level and quantitative summary of the domain knowledge. So we want to study special information that's contained in many neuroimaging articles. And one important difficulty is that although these studies rely on neuroimaging techniques, most of the time the actual images are not published. They are not made publicly available to protect the anonymity of participants or for many other reasons. So all that we get, all that is made public is text, a text report like a journal article. So the quantitative spatial information that was contained in the MRI images gets degraded to natural language and is now encoded in sentences like this one. So what we want to do in order to be able to perform statistics on their findings in brain space is to map back this text to the original brain space and reconstruct a brain image that we can associate with this study. And then we can pull many studies together and perform some statistical tests. So, for example, one way to use this would be to collect many articles that mention Huntington's disease and transform them into brain images, and then see how the spatial distribution of these brain images differs from some null distribution, to see which regions get mentioned very often when we study Huntington's disease. So I'll start by explaining how we perform this mapping from text to spatial densities over the brain, and then I'll present some of our results. I'll talk about the application I just mentioned about meta-analysis for pathologies, and finally I'll briefly discuss some ongoing work about using more advanced natural language processing techniques to alleviate the sparsity of text and the fact that the language used in cognitive neuroscience is sometimes redundant or ambiguous. So in order to find this mapping between text and concepts used in neuroscience and positions in the brain regions, we decided to take a supervised learning approach. So we are going to fit a statistical model to learn this mapping. So we started by collecting many journal articles about neuroscience, most of them about fMRI studies, and pre-process the text. And then we need to learn the mapping from this text to to the brain images they came from. Unfortunately, like I said before, usually we don't have these images. So the next best thing is peak activation coordinates that are often reported in tables in the articles. So usually when someone conducts a study using fMRI, they would, in the text of the study, describe the experiment they make. They will scan patients performing, for example, a certain task, and then they will find out which regions of their brain showed significant activations when they were performing the task. And these brain images are registered to a standard template, and there is a standard spatial referential of the brain, which enables researchers to give us, in tables in the article, coordinates in a standard referential where significant activations, called peak activations, occurred. So here we are told that what the interesting stuff that this article describes happened at these specific coordinates. And we can extract this, which gives us a set of positions in the brain, and then use density estimation to reconstruct an estimation of the density from which these positions came from. So a brain image that represents the content of this article in brain space. And this is what we are going to learn to predict. So to summarize, we obtain an article about functional neuroimaging, and we extract all the text, and then we vectorize it by counting word frequencies. So this gives us a vector whose dimension is the size of our vocabulary, which represents the text of the article. And we also find in tables peak activation coordinates. We perform the density estimation by convolving them with a Gaussian kernel, and this gives us the brain map that is the target that we associate with this article and that we will learn to predict. And then we simply fit a linear model because we want the coefficients of the model to be easy to interpret, and also because text is very sparse and high dimensional, so it makes sense to use simple models. Out of the many articles that we downloaded, we could extract peak activation coordinates from 14,000. So our data, this matrix has size 14,000 times a few thousands to a few hundred thousands for the vocabulary size. And then we, as I said, we fit linear regression to predict the activity of brain voxels given some vectorized representation of the text. Since what we are predicting is the probability density function of the activations that get reported, it makes sense to minimize the total variation between what we predict and the actual density. So that's not the spatial total variation we heard about before, but the total variation between two distributions, which in discrete spaces is the L1 norm of the error. And it looks a bit weird because of the kink of the L1 norm, but the dual of this problem is smooth with box constraint, and it can be solved very easily. So now that I presented the method that we use to learn the link between concepts and brain regions, I'm going to present some results. First of all, we need to have some metric to check that our predictions are actually better than chance and ideally better than previous methods. So since we predicted a density, we will look at the log likelihood of coordinates that are reported in the articles that are in the test set. It's a very natural thing to use, and we perform a cross-validation on our data set. So this enables us to obtain some scores that I show here. So, this is log likelihood, so more to the right here is better. And here I show different, each group corresponds to a different way to discretize the brain. Because we predict a density over the brain. In order to do this, we cut it up. We have to divide it into regions. So we can either use a grid of voxels or anatomical regions that have been segmented manually. And in this case, we would predict a probability for each region. So what we see here is that it's better to use a voxel-wise model and predict an activity for each voxel in the brain. And then we have some very naive baselines. One that uses the labels of the regions in anatomical atlases that have been segmented manually, and we see that it performs very poorly because when they write, people don't necessarily use the exact labels from any given atlas. And just some basic sanity check. So in the end, we see that the best results are obtained when this is a usual ridge regression, and this is the least absolute deviations, the robust regression. So the best results are obtained when we use robust regression and voxel-wise discretization of the brain. We can also compare to a tool that is called NeuroSynth, which is the closest existing method to what we're doing. And we can see that NeuroSynth performs significantly better than a naive baseline, and that the proposed methods perform even better at predicting the positions of peak activation coordinates in left-out articles. But what we're really interested in is, once we have checked that it can make predictions, is to look at the coefficients that are learned by the model, the mappings from words to positions in the brain that the model has learned. Because here, remember that these are word frequencies, so here each column corresponds to a word in our vocabulary, and we are predicting a density over all the voxels in the brain. So each row of the coefficient matrix maps one word to something that has the dimension of the brain. So it is the brain map that the model has learned to associate with a particular word. And for most words, we see that they look like what we would expect. So we can check on, for example, anatomical terms that they correspond to what we want. For example, this is the left amygdala. This is what it learns for amygdala and right amygdala. And for more psychological concepts like object recognition, we see, I guess, the fusiform face area, and for speech, we see the auditory cortex. So when we inspect the model, we see what we wanted to find. And then I said that this could be used to pull together many articles and try to find which regions are relevant when studying a particular pathology. So for example, if we want to learn which anatomical structures in the brain are important for Huntington's disease, this is a rare disease, and it's hardly discussed in the labeled corpus of 14,000 articles from which we could extract peak activation coordinates. So we don't have enough text about Huntington accompanied by brain images or peak activation coordinates to perform this kind of meta-analysis. But what we do have is a model that we have trained that maps text onto brain regions. And unlabeled text, for which we don't have images or activation coordinates, it is available in much larger quantities than labeled text. So we can download 100,000 articles. In these, we will find more that discuss Huntington's disease. And then using the model that we have trained on our labeled corpus, we can map this large amount of unlabeled articles to brain space, and then contrast the distributions of the predictions for articles that mention Huntington's against the mean distribution. And by performing this contrast, we highlight the putamen and the caudate nucleus that we know to be important when studying Huntington's disease. And we did the same thing with Parkinson, for example, and we see the motor cortex and the brainstem, which is also satisfying. I didn't try with schizophrenia, but maybe we'd see the ACC or something like that. Thank you. So this is the work we did to find simple mappings from terms to positions in the brain. But one difficulty and one limitation of this is that the occurrences of these terms are very correlated, and the representations of word frequencies for a document are very sparse. So even documents that talk about the same thing, if the authors come from different communities, they can use slightly different vocabularies, and these representations can be orthogonal even though the articles are actually quite similar. So what we need to do is to capture the similarities between these terms and their semantic relationships. And again, for this, we will leverage the fact that unlabeled text, plain text without any images, is available in very large quantities. So we can use this to learn good unsupervised representations for text, and then use these for the supervised learning that we want to do. So with this semi-supervised approach, we can reduce the sample complexity of learning mappings from text to brain regions. And it is well known that similar terms tend to occur in the same context and tend to occur together. So there is a lot of literature on learning good vector representations for words that capture their relationships. So these vectors are trained such that words that appear often together are associated with vectors that are highly correlated. So I think the most popular algorithm for this is, the most popular model is called the skip-gram negative sampling. And it's a simple matrix factorization model. And just as examples, I show here the words that have vectors that are very similar to that of visual cortex. And here the words that have vectors very similar to that of speech. And we see that they are all words that have similar meanings they all have similar meanings. So using these vector representations for these words, we can smooth the representations we use for document and facilitate the learning of mappings from text to positions in the brain. And this is what we are working on now. So we can download millions of articles about, for example, from PubMed, and learn good representations for words. Thanks. We select hyperparameters and check that these representations make sense by completing graphs, ontologies of the domain, or maybe graphs of links between Wikipedia pages, because if there is a link between two Wikipedia pages, usually the concepts are related. And then we use them to smooth the document representations we use. So a combination of our original representations for documents and these similarities provide a kernel on documents, and using kernel ridge regression we can do the kind of learning we were doing on smooth representations. So to sum up, we have put together a framework for translating text about neuroimaging to spatial densities over the brain. We have checked quantitatively that this enables us to make predictions on left-out data, and this has helped us to make choices about the models we use. It seems on a few examples that this enables us to pull together many studies for which we only have text and the imaging data is not available, and see where their findings agree and which brain regions are relevant for a certain context. And we are working on improving our representations for documents, and also next we want to release some data, so the coordinates, representations of the text, the brain maps that we learned, and provide a brain interface, a web interface, so that neuroscientists can have a look at and explore those results. And with that, thank you for your attention, and thank the organizing committee again.
Thank you, Jerome. Final round of questions.
So thank you for a very intriguing talk. I didn't know you could do this, so it's interesting. Could you then even use this to detect fake papers, like papers where where it's just kind of random text or things that are where figures are wrong and text are not well interpreted, bad interpretation or things like that? I guess you could, isn't it? Or
I guess. For a single paper, we don't expect a good prediction. So if we try to do this, we would detect many fake papers or many bad papers, because there's a lot of noise in both the output and the features. And it's more when we average this over many articles that we get interesting results.
Okay, thanks. Yeah.
On a completely different side, so from the fake papers to new knowledge, can you in some way infer new knowledge unexpected? Because then you have really a huge amount of data, so maybe you can find some correlations that were not planned at the very beginning. More than making a representation of what we know in some way.
I would hope so, and then we would need a closer interaction with the cognitive neuroscientists to figure out ways that this could be used to find new insights. But one example would be to find concepts that are very different in a semantic space, that are hardly discussed together or studied together, but that map onto very similar regions of the brain. Another way this could be used could be to provide priors or regions in interest in preparation for studies. People could write an abstract and then have a rough idea of which regions the literature suggests might be interesting. Or, this is something we need to explore more.
Thanks.
So how do you tackle the variability in reporting the same statistics but differently in different papers? That maybe they're just reporting the same thing, but they're reporting it in such a different manner. So are you missing out on those? Have you designed in the beginning of your task that this is going to be the standard design at which I'm going to report all of these values, and so you end up capturing only those papers? Or have you tackled this variability in reporting?
So in reporting, you mean in reporting the activation coordinates?
Yeah.
So usually they are in MNI. There are basically, I think, only two standard spaces, MNI and Taleyrac space, and very often they are in MNI. These spaces don't differ very much in many regions of the brain. So we try to look only for MNI coordinates, but if Taleyrac coordinates get included sometimes, I think this will add some noise, but there's already a lot of noise, but probably no systematic bias. And then for the ways that tables are organized or things are reported, then that's plain programming and trying to match templates to the tables that we find. And we have checked manually on many papers that the extraction process was working correctly.
Okay, thanks.
Are we okay? Let's thank Jerome once more.
Thank you.